You have a summer internship in a botany research lab at a university. The professor you are working for studies plant responses to various stimuli, including light, gravity, and contact. In order to familiarize yourself with some basic types of plant responses, the professor asks you to perform several experiments. In this interactive lab, you will test for plant reactions to light, gravity, and touch. Explore the lab to learn what is available for your investigation. Like all living things, plants respond to stimuli. Plants begin responding while still in the seed stage. Seeds will germinate only under the proper conditions. Growth in developing leaves and shoots is influenced by stimuli from the environment. Growth toward or away from a stimulus is called a tropism. Tropisms are caused by chemicals called hormones. Some stimuli that cause tropisms are light, gravity, and contact. This investigation studies only 3 types of responses. 2 of the responses observed in this investigation are tropisms, which means that they affect a plant's growth. Phototropism is the tendency of a plant to grow toward the light. Gravitropism is a plant's response to Earth's gravitational pull. Downward growth is positive gravitropism because the growth is in the direction that gravity pulls. Upward growth is negative gravitropism because it is growth against the force of gravity. The 3rd type of response observed in this investigation is known as a nastic movement. Nastic movement is the rapid response of a plant that does not involve growth. The rapid closing of a Venus flytrap's leaves is an example of nastic movement. The nastic movement of a mimosa plant is caused by changes in the water pressure surrounding the cells. Explore the lab to learn what is available for your investigation. You must click on each item in the checklist. Once you are ready to move on, click Procedure. 150 milliliter beaker with potting soil. Use the beaker filled with soil to grow the radish sprouts. Corn seeds. Use the corn seeds to observe how gravity affects root and shoot growth. Radish seeds. Use the radish seeds to observe how radish sprouts respond to light. Cardboard box. Use the cardboard box to control the direction from which light reaches the plants during your investigation. It has had the top and one of the sides cut off. Cover. Use the cover to simulate dark conditions. Aluminum foil caps. Use the foil caps to fit over the tips of the radish shoots. Water bottle. Use the water bottle to water the radish seeds daily, since water can evaporate from the potting soil. Wet cotton bunting. Use the wet cotton to hold the corn seeds in place in the petri dish while also supplying them with water. Wet filter paper. Use the wet filter paper to supply the water that the corn seeds need to germinate. Pack the corn seeds against the wet filter paper. Petri dish with cover. Use the petri dish to germinate the corn seeds. This will make it easier to observe the roots and shoots as they grow. Folded index cards. Use the index cards to support the petri dish in a vertical position during the investigation. The cards have been folded into L-shaped brackets. Mimosa plant. Use the mimosa plant to observe rapid responses, also called nastic movement. In this lab, you will observe how plants respond to stimuli. Open the lab notebook and record your predictions of how you think plants may respond to each of the following stimuli: light, gravity, and touch. Once you have recorded your predictions, click the arrow to advance to Step 2. In the first part of this investigation, you will observe the effect of light on the direction of radish plant growth. Drag the 4 radish seeds to the beaker to plant them a few millimeters below the surface of the soil. Drag the water bottle to the beaker to moisten the soil. Drag the cover to the beaker to grow radish plants in the dark for 3 days. Once 3 days have passed, click the arrow to advance to step 3. Drag the 2 aluminum caps over 2 of the shoots to cover their tips. Leave the remaining 2 shoots uncovered. Drag the cardboard box over the plants so that they get light from only one direction and grow radish plants for 4 days. Once 4 days have passed, click the arrow to advance to step 4. Click the aluminum caps to remove them. Observe the direction of shoot growth in the 4 plants. Open the lab notebook and record your observations. Once you have recorded your observations, click the arrow to advance to step 5. In the second part of this investigation, you will observe the effects of gravity on the direction of growth in corn. Drag the corn seeds to the Petri dish. Notice that they are arranged in a single row across the middle of the Petri dish, with the point of each seed facing a different direction: up, down, left, and right. Once you have placed the seeds in the Petri dish, click the arrow to advance to step 6. Drag the wet paper filter over the corn seeds. Drag the wet cotton to the Petri dish to fill it the rest of the way. Drag the cover to the Petri dish to cover it. Once the Petri dish is covered, drag the Petri dish to the index card stand. Drag the cover to the Petri dish to grow the corn seeds in the dark for 3 days. Once 3 days have passed, click the arrow to advance to step 8. Open the lab notebook and record your observations. Once you have recorded your observations, click the arrow to advance to step 9. In the third part of this investigation, you will observe rapid response or nastic movement in a mimosa plant. Drag the mouse over one of the mimosa plant's leaves. Open the lab notebook and record your observations. Once you have recorded your observations, click the arrow to advance to step 10. You are now ready to analyze your data and draw your conclusions. Open the lab notebook and answer the questions.